Hey guys, today we're going to be covering the cell cycle and how it's related to cancer. So to give an overview of this video, we'll start out by talking about what the cell cycle is and how it works with signals and checkpoints. From there we'll move into kinases and cycles that control it and how a disruption in that control can lead to cancer. The cell cycle is the life of a cell from the time it's first formed to its division into two daughter cells. The cell cycle accomplishes cell duplication, which can be used either to help an organism reproduce, grow and develop, or renew tissue. In the cell cycle, there are different phases, and we're going to focus on 4 of them, starting out with G1, which is the growth 1 phase. In this phase, the cell matures. The next phase, which is called the S phase, or the DNA replication phase, is where the DNA is replicated. The G2, or growth 2 phase, is the next phase, and the cell continues to get ready for cell division. And the final phase, which is the M phase, or the mitotic phase, is where the fully matured cell finally splits into two daughter cells that were identical to the parent cell. So this progression between phases in the cell cycle is only possible because of the checkpoints within the cell cycle. So checkpoints act as red and green lights for cells. If the checkpoint clears the cell, it will advance to the next phase, but if it blocks the cell, it goes into the G0 phase, or the growth 0 phase, where it can sit and wait until it's needed again and re-enter the cell cycle, or deteriorate. It's essential that these checkpoints are accurate because if cells ignore the checkpoints, or checkpoints didn't prevent defective cells from advancing through the cell cycle, abnormal cells would have the ability to divide freely, and that could lead to a cancerous tumor. The next way that the cell cycle is organized is through cyclins and cyclin-dependent kinases. Cyclins are groups of related proteins, and in most eukaryotes there are 4 types: G1, G1 over S, and M cyclins. Each cyclin helps carry out the event that its phase in the cell cycle is associated with. For example, the G1 cell cyclin encourages the cell to mature because in the G1 phase of the cell cycle, the cell's goal is to mature. So another example is in the S phase, or DNA replication phase. It's going to help replicate that DNA because the cell's purpose in that phase is to replicate its DNA. A cyclin-dependent kinase is a family of enzymes that is inactive until it's paired up with a cyclin. So the cyclin gives the enzymes a function. Once the CDK can function, it attaches phosphate groups to proteins to make them more or less active. Like the cyclins, they ensure that the cell is doing the right thing during each period of the cell cycle. So working together, the cyclins and cyclin-dependent kinases ensure that the cell doesn't have abnormalities because it's telling the cell what to do in each phase through specific signals. So if this control over the cell was lost, the cell could become cancerous because of errors in DNA replication. And without the guidance of the CDKs, they could develop abnormalities. There are also internal and external signals that make the cell cycle a highly regulated system. So the 2 growth factors that we're going to focus on are MPF, which is the mitosis promoting factor, and PDGF, or the platelet-derived growth factor. So both of these help cells to divide, but the mitosis promoting factor specifically works on the nuclear membrane during the M phase of the cell cycle. So in the mitosis, promoting factor does is helps break down that nuclear membrane by adding phosphate groups to its proteins that weaken it. And that eventually allows that nuclear membrane to divide and for the two daughter cells to be fully formed. So PDGF lets other cells divide, and it signals those cells by binding to their cell receptors and phosphorylating it. So the phosphorylation of the cell sends a signal for the cell to divide. When both of these growth factors are depleted, the cells know to stop dividing. So if the growth factors don't function properly due to an abnormality in the cancer cell, it could grow and reproduce without the growth factors, could even use its own to bypass the checkpoints in the cell cycle. And that can lead to cancer as well. Our last topic is that cancer results from disruption in cell cycle control. So cancer cells have abnormalities, generally from a mutation during DNA replication, that makes the cell cycle control faulty. Since the cell cycle cannot properly function, the cancer cell can bypass apoptosis, which is the suicide of a cell triggered by a checkpoint in the cell cycle. So in apoptosis, a cell is breaking itself down through the use of self-destructive enzymes, and it receives a signal to destroy itself because it does great harm to the organism if it replicates instead of self-destructing. So if the cancer cell gets past apoptosis and sneaks by the checkpoints in a cell, the immune system still tries to destroy it, but if it gets by the immune system, it's free to replicate, and that's when you have the formation of a tumor. So sometimes the tumor isn't strong enough to survive outside of its tissue that it's already occupying, so it doesn't spread, and that's called a benign tumor. So benign tumors, they stay in their location. They aren't taking over healthy tissue and making it cancerous, and they're removed through surgery. But when the cancer cells do spread, it's called a malignant tumor. And the malignant tumor causes these cancer cells to take over healthy tissue and turn it into cancerous tissue. So this is why it's so important for cell checkpoints to work properly. Because if a cancer cell is not signaled to destroy itself or to stop production, it can replicate. And if the control of the cell cycle is lost, it's very hard to prevent cancer.